Hi, everyone. My name is Dr. Matt Yost. I'm an agroclimate extension specialist at Utah State University, and I'm happy to be here with you today, albeit remotely. I apologize that I was not able to come in person. When Garrett invited me to come, I was very excited to come and feel bad that I was not able to make it in person. I've been previously asked to host a meeting of soil fertility experts and faculty from across the West Monday and Tuesday next week. So was not able to get out of that assignment. So I happy to be with you virtually today, and was asked to kind of prime a discussion on soil microbiology. And so that's what I'd like to do today. This is a difficult task to do this in about 10 minutes. And so I'm going to do my best and try to talk about this in a practical way. I was raised on a dairy farm in Burley, Idaho, and have always liked to think practically and in a way that farmers might think as you're trying to make decisions on the farm. So that's what we're going to try to do today. If I can get this to work, here we go. So we're just going to go through a few questions together about microbiology in the soil. So first question, what lives in your soil or my soil? A lot of things. There's a whole system, a whole food web with lots of pieces and parts. Today we're going to talk about two of the big ones, and that's just bacteria and fungi. These are a part of what we would generally call a microbe or a microorganism in the soil. So bacteria and fungi, I want to talk about these because they are, if you look at their number and their mass in the soil, they represent the majority. So most of you have probably heard that in a teaspoon of soil, we have a lot of life, right? Up to millions and even billions of bacteria and fungi in just one teaspoon. Pretty incredible. What's even more interesting, and I think an easier way to think about this is when it's converted to a per acre basis. And so on a per acre basis, if you look at if you tried to count the bacteria or fungi, you get into these um, quintazillion or just really high numbers, which are hard to understand and grasp. But if you look at the weight, so there's a lot. There's a lot of fungi and a lot of bacteria. If you look at their weight, um, that's an easier thing to understand. So fungi, anywhere from 1,000 to 15,000 pounds per acre in the top 6 inches of soil. So up to roughly 6, 7 tons per acre of fungi in your soil. Bacteria, or actinomycetes, also another form of bacteria, can be up to about 5,000 pounds. So um, roughly 2.5 tons per acre of, of bacteria in your soil. So, some people talk about this, and you've probably heard this, that microorganisms are like livestock, right? Underground in the soil, and are significant. So, 2 to 7 tons per acre of organisms living in your soil. So, just a few more things about bacteria and fungi. There's bacteria, just thousands of known species, and that's known. Many that we don't know and are learning. They change rapidly. Some bacteria can reproduce in as little as 30 minutes in ideal conditions. So they're just always changing, depending on the conditions in the soil. Fungi, a few characteristics of them, they are important because they produce long strings of cells called hyphae, which really help improve structure in the soil and also help plants, but they connect with plants and help plant roots um, better uptake nutrients and water. They also, as a part of that, they provide a glomalin, which is similar to glue, like a glue-like substance that helps with soil aggregation. And we know and we understand that aggregation is very important for lots of things in soil functions. Okay, so we know there's a lot. Now, what do they do in our soil? I'm going to try to keep this really simple because it can get complex fast. Some people describe bacteria and fungi as the first responders in the soil. They're the first things that get to start working on crop residue, organic materials that we add in our soil, dead roots. They're kind of the first thing that start working on them. And here are some of the things that they do and the reason why they're so important in the soil. They're decomposers. Primary function of microbes is to decompose, to eat. And when they do that, they create organic matter. They make nitrogen available. They improve water filtration. They can improve soil aggregation. Some bacteria can fix nitrogen. They can also compete. They're constant competition with each other and also with pathogens. So in some cases can reduce disease and other negative effects of pathogens. Can also help enhance root nutrient and water uptake and can degrade pesticides and other pollutants. So we know they're important. We know there's a lot of them. How do I measure them in my soil? So there's a few ways to do this. Probably the most well known and available, something called a phospholipid fatty acid profile or analysis. This basically looks at these fatty acids, which are specific to different types of fungi and bacteria. This is a test that some labs offer, not a lot. One of the most common is Ward Laboratories in Nebraska. They will conduct a PLFA test for about $80. And this is what a report would look like. And so you can see here that what they measure is kind of an estimated total biomass of everything, every microorganism in the soil. And then they also get an estimate of diversity. How diverse are the microorganisms? And they've rated them there. You can see from very poor to excellent. If you've got a lot, and they're highly diverse, that's an excellent rating. You'll also notice here that they can go through and estimate different types. So they can estimate total bacteria, kind of its biomass, total fungi, different types of fungi and different types of bacteria, protozoa, and then a group of other organisms that can't be separated. This is just a portion of the report. If you go and look on Ward Laboratories webpage, you can see that they go through and describe kind of what each measurement is and what good ratings might be for each of those categories. There's another similar analysis called FAME. It's another type of fatty acid analysis, not as widely available to producers, say more available in the research community. The more crude but less expensive test is called soil respiration. It's also known by other names like a CO2 burst or flush or Solvita. They're all kind of the same thing. Basically, what's being done here is you take a soil sample, you dry it, and then you wet it. And for 24 hours after you wet it, you measure the CO2 that's released. Some people describe this as the soil microbes breathing, and you're kind of measuring how much they're breathing. And so it's an estimate of potential activity of the microbes. So the more CO2 that's released means that there's more potential microbial activity. There's more microbe biomass in that soil. And so this is just a summary here of what a report might look like for Ward Laboratories. they offer soil respiration, as do many other labs. Um, and they say that most agricultural soils that they see are usually below about 200 parts per million carbon dioxide that they measure in the soil, and they've gone through and kind of described what different rankings might be for different concentrations of CO2 release from the soil. So this is more crude, but a quick way to get an idea of how much biomass you might have of microbes and how active they might be. Okay, probably the most important question today is, okay, how can I improve the health of microbes in my soil? Here's a few ideas. One, which most of you are probably already doing, where this is a direct seed meeting, but minimizing or eliminating tillage is a great way to improve the health of bacteria and fungi. This is particularly beneficial to fungi. A study in Nebraska found that when you till, it reduces fungi populations. Not indefinitely, but for a time. Bacteria are more adaptive. They can change quite rapidly and can adapt to tillage, but fungi are the ones that generally are hurt more with tillage. Constantly adding organic matter with low C to N ratios, if you can, is a great way. It feeds them. You're basically giving your microbes food. The other thing that you can do is just grow healthy plants with healthy roots. Roots are critical for bacteria and fungi. That's where most of them live, is right around the roots. This one's tricky, but if you can, pay attention to soil temperature. Microbes like it warm and wet. That's how they consume more and reproduce more under those conditions. So the best temperature for activity, maximum activity, is about 85 degrees in the soil. If you get outside of about 30 to 110 degrees, there's very little or no activity. And so, there's only so much that can be done here, but just managing residue and cover to try to keep soil cool and inside that range will help promote activity. Pay attention to your pH, which we normally do anyway for nutrient uptake and other reasons, but ideal range here is between 6 and 7.2. That's what nitrogen fixing bacteria like, and that's beneficial for other reasons too. Manage salinity. Salinity gets out of hand. It's bad for the crop. It's also bad for the microbes. Irrigation. Just avoid over irrigation and stay near field capacity. And in some soils, they suggest that even a little bit below field capacity is ideal for microbes. This last one, inoculate soil with more microbes or stimulants. This one's tricky. There's a lot of marketing that happens around this idea, especially recently. Lots of products out there on the market that either microbes or stimulants. In some cases, they can work, but in a lot of cases, they just don't do much. Here's an example. So in Nebraska, they tested 17 products that, really common products on the market, and it's 76 farm trials across their state, and only 8% of them had a yield response to any of these stimulants or microbe applications. It can happen, but in most cases, if you've got a healthy microbes in your soil and you're adding organic matter and you're irrigating correctly and you're managing these other things correctly, you're going to have a healthy microbe population. And adding stimulants or more microbes in a lot of cases are not going to provide a lot of benefits. Okay, so now I'll turn the time to you guys to have some discussion, and two questions that I thought you might be interested in discussing is one, just what could you do to start to look at your microbes in your soil? So looking at some of the tests and measuring what you have, the current amount and diversity of microbes that might give you a baseline as to what your microbe health looks like, which will then help you assess how to improve that. The second one here is to maybe discuss what practices that you could do on your farm or on the farms of those that you advise to economically and feasibly improve the health of your microbes to enhance your wheat production. That's all I have. Hope you have a great discussion, and look forward to connecting in the future. Thanks.